Right, we're going to go into fairly detailed and reasonably long expose now. I will try and get the transcript up of this fairly quickly because it's a lot to take in, a lot of technical detail and a lot of stuff that we don't really know a vast amount about. But this is another phase of the fake COVID virus and its future, or lack of a future, this dabble in technology can bring. A gene drive is an existing technology of genetic engineering that is able to propagate a particular suite of genes throughout a population by altering the probability that a specific allele, ALLELE, will be transmitted to offspring instead of the Mendelian 50% probability. And the application is particularly suited for creating an irreversible species extinction. Their words, their document. The US Defense Advanced Research Projects Agency, DARPA, has been given approximately $100 million for gene drive research, making them the largest single funder of gene drive research on the planet. The top secretive level group, Jason Group, of military advisors produced a classified study on gene drive in 2017, reflecting an extremely high level of interest and activity by other sections of the US military and intelligence communities. The Bill and Melinda Gates Foundation paid a PR firm $1.6 million to secretly stack key UN advisory processes with gene drive-friendly scientists. Now, gene drives can arise through a variety of mechanisms. They have been proposed to provide an effective means of genetically modifying specific populations and entire species. The technique can employ adding, deleting, disrupting, or modifying genes. Proposed applications include exterminating insects that carry pathogens, most notably mosquitoes that transmit malaria, dengue, and Zika pathogens, controlling invasive species, or eliminating herbicide or pesticide resistance. But as with any potential powerful technique, gene drives can be misused in a variety of ways or induced unintended consequences. For example, a gene drive intended to affect only a local population might spread across an entire species. Gene drives used to eradicate populations of invasive species in their non-native habitats may have consequences for the population of the species as a whole, even in its native habitat. An accidental return of individuals of the species to its original habitats through natural migration, environmental disruption, for example storms or floods, accidental human transportation, or purposeful relocation could unintentionally drive the species to extinction if the relocated individuals carried harmful gene drives. It is very much easier to kill or sterilize a plant using gene editing than it is to make a herbicide or insect resistant, according to Guy Reeves, who is an expert in GM insects at the Max Planck Institute for Evolutionary Biology, the very same company that we brought up in From Russia with Love 4, was it? 4 or 5 anyway. Now, gene drives can be built from many naturally occurring selfish genetic elements that use a variety of molecular mechanisms, and these naturally occurring mechanisms induce similar segregation distortion in the wild, arising when alleles evolve molecular mechanisms that give them a transmission chance greater than the normal 50%. And most gene drives have been developed in insects, most notably mosquitoes, as a way to control insect-borne pathogens. Recent developments designed gene drives directly in viruses. Recent developments designed gene drives directly in viruses, notably the herpes viruses. These viral gene drives can propagate a modification into the population of viruses and aim to reduce the infectivity of the virus. And in December of 2017, documents released under the US Freedom of Information Act showed that the military research agency DARPA had invested $100 million in gene drive research, which makes them the likeliest and largest single funder of this type of research. The documents also reveal that DARPA either funds or coordinates with almost all major players working on gene drive development, as well as the key holders of patents on CRISPR gene editing technology. We've covered that before, CRISPR. Given that DARPA is a military agency, we find it surprising that the obvious and concerning dual-use aspects of this research have received so little attention, according to Felix Beck, who is a lawyer at the University of Freiburg. Now, the secret top-level Jason group of military advisors produced a classified study on gene drive in 2017, reflecting an extremely high level of interest and activity by other sections of the US military and intelligence community. And emails show that the Jason study was initiated with a 2-day meeting of a select group of invited gene drive researchers in June 2017. Think of the timeline. And at the meeting, the Vice President of Global Biotechnology for Monsanto gave a presentation on crop science and gene drives. And IARPA, an organization within the Office of the Director of National Intelligence, has also expressed interest in funding gene drive work. A scientist involved describes IARPA as basically the intelligence agency's version of DARPA, which may be more frightening, he said. And like we said earlier, the Bill and Melinda Gates paid a PR firm $1.6 million to stack the UN advisory process with gene drive friendly scientists, and then recruited ostensibly independent academics and public officials into a private collaboration to counteract proposed regulations and to resist calls by scientists and conservationists for an international moratorium, all kept in-house. Now, the group called Target Malaria, or the program, which is a project funded again by Bill and Melinda Gates Foundation, invested $75 million in gene drive technology. And the foundation originally estimated the technology to be ready for field use by 2029 somewhere in Africa. However, in 2016, Gates changed this estimate to sometime within the following 2 years. So, this gene editing technology was ready in 2018 then. Are you connecting the dots now? Because Target Malaria hopes to deploy their gene drives in African countries, they have been at pains to emphasize independence from military agendas. However, Target Malaria's Andrea Crisanti, working at Imperial College, is also a lead grantee or subcontractor for DARPA's Safe Genes project, having confirmed he had been hired by DARPA on a $2.5 million contract. Imperial College London, we also covered him from Russia with Love, Plus I think, has been a pioneer in gene drive research with DARPA funding, and in trials 2016 through 18, scientists succeeded in destroying a population of mosquitoes in a lab by introducing a genetic mutation that spread through the population and eventually sterilized all of the mosquitoes. Sterilized being the operative word. May not be mosquitoes. In previous experiments, mosquitoes had small random mutations that immunized them against the gene drive, and the Imperial College scientists created a gene drive that did not fall prey to this type of resistance. Since it can never be more than double in frequency with each generation, a gene drive introduced in a single individual typically requires dozens of generations to affect a substantial fraction of a population. Alternatively, releasing drive containing organisms in sufficient numbers can affect the rest within a few generations. For instance, by introducing it in every 1,000 individual, it only takes 12 to 15 generations to be present in all individuals. And it said whether a gene drive will ultimately become fixed in a population and at which speed depends on its effects on individuals' fitness, on the rate of allele conversion, which is cell, and on the population structure. In a well-mixed population and with realistic allele conversion frequencies, 90%, population genetics predicts that gene drives get fixed for selection coefficient smaller than 0.3. For the benefit of John Hopkins, that is a 0 with a tiny dot at the bottom and a number 3. In other words, gene drives can be used to spread modifications as long as reproductive success is not reduced by more than 30%. And in contrast with normal genes, which can only spread across large populations if they increase fitness. Why is fitness a key? Because the strategy usually relies on the simultaneous presence of an unmodified and a gene drive allele in the same cell nucleus. And it had generally been assumed that a gene drive could be engineered in sexually reproducing organisms, excluding bacteria and viruses. However, technological breakthroughs have enabled the design of a gene drive strategy that doesn't involve sexual reproduction, but relies on co-infection of a given cell by a naturally occurring and an engineered virus. A gene drive strategy that doesn't involve sexual reproduction but relies on co-infection of a given cell by a naturally occurring and an engineered virus. That line screams out right there, does it not? This virus, in my opinion, is to test large quantities of people and insert gene-altering technology. But who decides the parameters of this program? Who made them God? Or source? Where is source in these matters? Is he too busy telling one person to create an internet TV channel? And the fact that it's being run by military top brass personnel who have almost zero empathy, one has to question, where is this all going? Now, upon co-infection, the unmodified genome is cut and repaired by homologous recombination, producing new gene drive viruses that can progressively replace the naturally occurring population. And in cell culture experiments, it was shown that a viral gene drive can spread into the viral population and strongly reduce the infectivity of the virus, which opens up novel, there's that word again, remember the change from coronavirus to novel or novel therapeutic strategies against the herpes viruses, it said. Because gene drives propagate by replacing other alleles that contain a cutting site and the corresponding homologies. Their application has mostly been limited to sexually reproducing species because they are diploid or polyploid and alleles are mixed at each generation. But as a side effect, inbreeding could in principle be an escape mechanism. But the extent to which this can happen in practice is difficult to evaluate. So to avoid getting this gene editing technology, you have to do inbreeding. So the escape mechanism is to be like all the royalty and elite circles, following their Bible cousins and the Egyptian hierarchy of let's keep it all in the family then. No wonder incest is mentioned 54 times in the Bible. Now, due to the number of generations required for a gene drive to affect an entire population, the time to universality varies according to the reproductive cycles of each species, and it may require under a year for some invertebrates, but centuries for organisms with years-long intervals between birth and sexual maturity, such as humans. Hence, this technology is of most use in fast reproducing species. Well, given they've already hammered the ability for the white people to reproduce as quickly as they used to, One would suggest this was for something else. Other problematic issues highlighted by researchers include mutations. A mutation could happen mid-drive, which has the potential to allow unwanted traits to ride along. Host possession. Escape, crossbreeding, or gene flow potentially allow a drive to move beyond its target population. Ecological impacts. Even when new traits' direct impact on a target is understood, the drive may have side effects on the surroundings. And the Broad Institute of MIT and Harvard, no surprise, they're involved also, added gene drives to a list of uses of gene editing technology it doesn't think companies should pursue. Yes, because they're all going to make money on it themselves and play God is why. Gene drives affect all future generations and represent the possibility of a larger change in a living species than has been possible before. In December of 2015, scientists of major world academies called for a moratorium on inheritable human genome edits that would affect the germline, including those related to CRISPR-Cas9 technologies, but supported continued basic research and gene editing that would not affect future generations. In February of 2016, British scientists were given permission by regulators to genetically modify human embryos by using CRISPR-Cas9 and related techniques on condition that the embryos were destroyed in 7 days. And if you believe that, you'll believe anything. Why were they even doing it to begin with? In June of 2016, the US National Academy of Sciences, Engineering and Medicine released a report on their recommendations for responsible conduct of gene drives. Models suggest that the extinction-oriented gene drives will wipe out target species, and that drives could reach populations beyond the target given minimal connectivity between them. Kevin Esvelt stated that an open conversation was needed around the safety of gene drives, and in our view it is wise to assume that invasive and self-propagating gene drive systems are likely to spread to every population of the target species throughout the world. Accordingly, they should only be built to combat true plagues such as malaria, for which we have a few adequate countermeasures and that offer a realistic path towards an international agreement to deploy among all affected nations. And he moved to an open model for his own research on using gene drive to eradicate Lyme disease, which in and of itself was a biological weapon which was released by accident, yes, US military again, in Nantucket and also Martha's Vineyard, which is common for US presidents to attend, is it not? X as well. Esvelt and colleagues suggested that CRISPR could be used to save endangered wildlife from extinction, and later retracted his support for the idea, except for extremely hazardous populations such as malaria-carrying mosquitoes and isolated islands that would prevent the drive from spreading beyond the targeted area. Austin Burt, an evolutionary geneticist at Imperial College London, introduced the possibility of conducting gene drives based on natural homing endonuclease selfish genetic elements in 2003. And researchers have already shown that such genes could act selfishly to spread rapidly over successive generations. Burt suggested that gene drives might be used to prevent a mosquito population from transmitting the malaria parasite or to crash a mosquito population. Now, gene drives based on homing endonuclease have been demonstrated in the laboratory in transgenic populations of mosquitoes and fruit flies. However, homing endonucleases are sequence specific, and altering their specificity to target other sequences of interest remains a major challenge, they said, and the possible applications of gene drive remained limited until the discovery of CRISPR and associated RNA-guided endonuclease such as Cas9 and CPF1. Gene drive and associated RNA, like the vaccine, mode RNA. In June 2014, the World Health Organization Special Program for Research and Training in Tropical Diseases issued guidelines for evaluating genetically modified mosquitoes. In 2013, the European Food Safety Authority issued a protocol for environmental assessment of all genetically modified organisms. In sexually reproducing species, most genes are present in 2 copies, which can be the same or different alleles, either one of which has a 50% chance of passing to a descendant. By biasing the inheritance of particular altered genes, synthetic gene drives could spread alterations throughout a population. At the molecular level, an endonuclease gene drive works by cutting a chromosome at a specific site that does not encode the drive, including the cell, to repair the damage by copying the drive sequence onto the damaged chromosome. This is exactly what Bill Gates was describing with the RNA coding. Now, the cell then has 2 copies of the drive sequence, or the code. And the method derives from genome editing techniques, and as a result, the gene drive insertion in the genome will reoccur in each organism that inherits 1 copy of the modification and 1 copy of the wild-type gene. If the gene drive is already present in the egg cell when received from one parent, all the gametes of the individual will carry the gene drive instead of 50% in the case of a normal gene. And scientists have designed multiple strategies to maintain control over gene drives. The Drosophila drive requires at least 1,000 of insects for the drive to begin. A few individuals escaping the target region would be unlikely to spread the drive. Now, in 2020, researchers reported the development of 2 active guide RNA-only elements that, according to their study, may enable halting or deleting gene drives introduced into populations in the wild with CRISPR-Cas9 gene editing. And the paper's senior author cautions that the 2 neutralizing systems they demonstrated in cage trials should not be used with a false sense of security for field-implemented gene drives. CRISPR is a DNA editing method that makes genetic engineering faster, easier, and more efficient. And the approach involves expressing an RNA-guided endonuclease, such as Cas9, along with the guide RNAs directing it to a particular sequence to be edited. Coding. And when the endonuclease cuts the target sequence, the cell repairs the damage by replacing the original sequence with an homologous DNA. They've cut out the DNA and replaced it with the code DNA. That's what that's telling you. By introducing an additional template with appropriate homologs, an endonuclease can be used to delete, add, or modify genes in an unprecedentedly simple manner. As of 2014, it had been tested in cells of 20 different species, including humans. In many of the species, the edits modified the organism's germline, allowing them to be inherited, tagged, in other words. In 2014, Esvelt and co-workers first suggested that the CRISPR-Cas9 might be used to build endonuclease gene drives. In 2015, researchers published successful engineering of CRISPR-based gene drives in Saccharomyces, Drosophila, and also mosquitoes. All 4 studies demonstrated efficient inheritance distortion over successive generations, with one study demonstrating the spread of a gene drive into a laboratory populations. And drive-resistant alleles were expected to arise for each of the described gene drives. However, this could be delayed or prevented by targeting highly conserved sites at which resistance is expected to have a severe fitness cost. Now, because of CRISPR's targeting flexibility, gene drives could theoretically be used to engineer almost any trait. What a control mechanism that is. Unlike previous designs, they could be tailored to block the evolution of drive resistance in the target population by targeting multiple sequences within appropriate genes. CRISPR could permit a variety of gene drive architectures intended to control rather than crash populations. Gene drives have 2 main classes of application, which have implications of different significance. Introduce a genetic modification in laboratory populations, and once a strain or line carrying the gene drive has been produced, the drive can be passed to any other line just by mating. And here the gene drive is used to achieve much more easily a task that could be accomplished with other techniques. Introduce a genetic modification in wild populations. Gene drives constitute a major development that makes possible previously unattainable changes. And because of their unprecedented potential risk, safeguard mechanisms have been proposed and also tested. And one possible application is to genetically modify mosquitoes and other disease vectors so that they cannot transmit diseases such as malaria and dengue fever. And researchers have claimed that by applying the technique to 1% of the wild population of mosquitoes that they could eradicate malaria within a year. Perhaps they could eradicate the 1% within a year. Wouldn't that be useful? A gene drive could be used to eliminate invasive species and has, for example, been proposed as a way to eliminate invasive species in New Zealand. And gene drives for biodiversity conservation purposes are being explored as part of the genetic biocontrol of invasive rodents program because they offer the potential for reduced risk to target non-species and reduce costs when compared to traditional invasive species removal techniques. But given the risks of such an approach described below, the G-Bird Partnership is committed to a deliberate stepwise process that will only proceed with public alignment. That will be a first, as recommended by the world's leading gene drive researchers from the Australian and also the US National Academy of Sciences and many others. Now, a wider outreach network for gene drive research exists to raise awareness of the value of gene drive research for the public good. Yeah. Some scientists are concerned about the technique, fearing it could spread and wipe out species in native habitats, and the gene could mutate, potentially causing unforeseen problems, as could any gene. And many non-native species can hybridize with native species such that a gene drive afflicting a non-native plant or animal that hybridizes, what an interesting word that is, with a native species could doom the native species. And many non-native species have naturalized into their new environment so well that crops and or native species have adapted to depend on them. Now, the name of it, and they are real. The Predator Free 2050 project is in New Zealand government program. The Predator Free 2050 project. To completely eliminate 8 invasive mammalian predator species, including rats, short-tailed weasels and possums from the country by 2050. And the project was first announced in 2016 by New Zealand's Prime Minister then, John Key, and in January 2017 it was announced that gene drives would be used in the effort. In 2017, one group in Australia and another in Texas released preliminary research into creating daughterless mice using gene drives in mammals. And in 2017 at the University of California, Riverside developed a gene drive to attack the invasive spot-wing Drosophila, which is a type of fruit fly native to Asia that is costing California's cherry farms $700 million per year because of its tail's razor-edged ovipositor that destroys unblemished fruit. Now, the primary alternative control strategy involves the use of insecticides called pyrethroids that kills almost all insects that it contacts. Now, the transhumanist philosopher David Pearce has advocated for using CRISPR-based gene drives to reduce the suffering of wild animals. It depends on what you define as a wild animal. The transhumanist philosopher. And Kevin Esvelt, an American biologist who has helped develop gene-drive technology, has argued that there is a moral case for the elimination of the New World screwworm through such technologies because of the immense suffering that infested wild animals experience when they are eaten alive. That is the classic seducement program put forward for helping something like wildlife, insects, and disease. When these very people who run these programs have been involved in destroying ecosystems, mass extinction of plants, insects, animals, and mammals, destroyed crops with their bombs, munitions, and destroyed whole other sections with their bio-warfare experiments. And we are supposed to trust these people with this technology? THI put forward a solution, was it 2 to 3 years ago, about creating a committee of we the people to go with various types of scientists and analyze every piece of technology. It is a must for this planet to go forward. Every single research centre connected to military or agencies needs to be shut down, and every piece of technology and research needs to be evaluated of, is this beneficial to humanity, yes or no? That is not based on financial gain, fame, or contracts. Is it beneficial for the people and the planet? If no, then it has to be deleted. Now, we will cover the implications of this and more in an up-and-coming must-listen-to show. We'll be back shortly.